This is problem set number 4 for Biochemistry Free-for-All, and also the problem set used by Kevin Ahern's Biochemistry 450 class at Oregon State University. In this problem, I'll be talking about the structures of proteins. The first question asks, what two parts of an amino acid are joined together in making a peptide bond? The answer to that question, of course, is that the alpha amine of one amino acid is joined to the alpha carboxyl of another amino acid. We remember that the direction of synthesis of a protein within a ribosome occurs starting with the amino end and moving in the direction of the carboxyl end. That means, therefore, that for the very first amino acid joined to the second amino acid, that is the alpha carboxyl of the first amino acid that is joined to the alpha amine of the second amino acid. That leaves the alpha carboxyl on the first amino acid free and the alpha carboxyl of the second amino acid free. And when I say free, I mean not joined to any other amino acids. It turns out that all proteins, as a consequence of that, always have a free alpha amino group corresponding to the very first amino acid that the synthesis started with, and a single alpha carboxyl group that corresponds to the last amino acid put in. All of the other alpha amine and alpha carboxyl groups are joined together in peptide bonds to make the polypeptide chain. What parts of a polypeptide backbone behave as if it were a double bond? And the answer here, of course, is in fact the peptide bond. The peptide bond has what we call a resonance structure, where part of that resonance structure is, in fact, a double bonded molecule. So because of that, the peptide bond behaves as if it were double bonded, and that double bonded molecule will not rotate. So the peptide bond acts as if it's a fairly rigid structure, just like it were, as if it were a full double bond. Number 3 says, name the primary forces stabilizing alpha helices and beta strands. The primary forces that stabilize these are hydrogen bonds. In the case of the alpha helix, the hydrogen bonds are commonly found between the various amino acids that are spaced about 4 amino acids apart, because the rotation of the alpha helix occurs with a periodicity of about 3.6 amino acids per turn. Number 4 says, how does a supersecondary structure differ from a secondary structure? Now, supersecondary structure is a term that I use in BB 450 to refer to organization of secondary structures into higher order structures. So, for example, if I take a beta strand, which is simply that folded pleat that we see, what I described in class as a flattened alpha helix, that is taking an alpha helix that's a 3-dimensional structure and flattening it into a 2-dimensional form. If I take a bunch of those beta strands and I align them with each other, I can make what's called a beta sheet. So to sketch out what I'm talking about here, so I have a single beta strand that has a structure like this. When I start putting those together into higher order structures, I create what I refer to as a super secondary structure. The more of those that I add, the bigger the structure, of course, and that's a super secondary. Each alpha helix, each of the beta strands in each case is a secondary structure, but the sum of all of them put together, like here, is that of a super secondary structure. And by the way, these are all joined to each other, so they're all part of the same protein. The question number 5 says, name 3 fibrous proteins described in class. And in fact, I named exactly 3 fibrous proteins in class. Collagen I described as the protein that was the most abundant protein in our body. It's very important in connective tissue, and I described it as the glue that holds us together. Collagen is a helix of helices, and that's a very interesting thing. Collagen fiber has 3 helices, and those 3 helices are intertwined to make a braided kind of a structure. And that's very important for the strength and flexibility that collagen has. The second fibrous protein that I described in class is keratin. Keratin proteins are very important in things like hair or nails or the horns of animals. Another important fibrous protein that I described in class is fibrin. Fibrin is a protein that is found abundantly in silk. And fibrin is noteworthy because it has a lot of beta strands arranged in a supersecondary structure to form sheets. Number 6 says name 2 modified amino acids in collagen and the role each plays in the protein's structure. Well, first of all, the most abundant modified amino acid we find in collagen is that of hydroxyproline. And the second modified amino acid we find in collagen is hydroxylysine. Now, in both cases, the protein is made originally with proline in one case and lysine in the other case, and the hydroxyl groups get placed onto those amino acids as a result of a post-translational modification. So a post-translational modification occurs when an amino acid in a protein is chemically modified after the protein is made. So collagen is chemically modified by addition of hydroxyl groups to prolines and also to lysines. Now, not every proline gets a hydroxyl group, and it's kind of a complicated process. But suffice it to say that the addition of hydroxyl groups to proline and to lysine are very important for the structure of the overall protein. The addition of hydroxyls to proline causes collagen to be able to be stable at the temperature of the human body. Without those hydroxyl groups being added to proline, collagen is not stable, and the collagen itself will be very weak. The lysine hydroxylation is very important for binding the strands together because the hydroxyl groups of lysine can be joined together between individual strands to give strength to the collagen. So hydroxylation of lysine is important for the strength. Hydroxylation of proline is important for the stability at the temperature of the body. The combination of those two allows our body to have a collagen that's functional and that's also strong as necessary. Now I note that the hydroxylation of both proline and of lysine requires vitamin C. In the absence of vitamin C, a person who's deficient in vitamin C, for example, very weak collagen is produced, and the person develops a condition known as scurvy. This can be fatal, of course, if severe, because the inability of collagen to be stable and to help hold together the tissues of the body could have some pretty drastic ramifications. It's treated very easily, however, by having abundant vitamin C in the diet. Question number 7, I've already answered. Why is vitamin C essential for a strong collagen? So I won't go through that again. Question number 8, you discover a new protein that has numerous alternating regions of high hydropathy interspersed with short regions of high hydrophilicity. Describe a likely structure of such a protein. Well, we can imagine a lot of different possible structures, but I want to get you to see one that I've actually shown you in class and to think about that. So I'm going to draw that schematically in this figure. I start by describing to you a membrane. Now, I showed you a little bit of a membrane in class, and membranes are interesting in that they have a lipid bilayer nature to them. They have long nonpolar tails on the inner part of the bilayer, and they have polar heads on the exterior of the bilayer. So we could say, for example, this is inside of a cell, and we could say this is outside of a cell. And we have a lipid bilayer of all of these things here. Well, of course, cells are about 70% water, so we have water surrounding this lipid bilayer. There's water out here, there's water out here, and there's essentially no water inside of here. Well, if we have a protein that resides in a lipid bilayer, and I'm going to draw the bilayer separately over here again without the labels. A protein that exists in this environment has parts of it that will be touching water, parts of it that will be touching very nonpolar things, the part of the tails that are being held there, and finally, another part that's down here touching water. Well, a protein that's what we call a transmembrane protein exists in all of these environments. So the question says that it has alternating regions of high hydropathy and high hydrophilicity. Well, hydropathy, of course, refers to being hydrophobic, meaning it doesn't like water, and that hydrophilic portions of a protein are those that interact easily with water. Well, if I have such a protein, I'm going to label it in red in this figure. If I have such a protein, I could imagine that it might wind through a lipid bilayer something like this. Doing that, I recognize that different parts of the protein will have different properties. This will be, I'm going to put HF for hydrophilic, cheating with my spelling there. Hydrophobic, meaning HPH. And then hydrophilic, again down here. Because this part's touching water, this part is not touching water, this part is touching water. So we could see that if we had this protein embedded in a membrane, that its structure would alternate between having hydrophilic, hydrophobic, hydrophilic, hydrophobic, hydrophilic, hydrophobic regions. This is actually one of the ways that we can look at the sequence of a protein and sometimes get an idea about whether or not this is a membrane protein or if it's a protein that in fact instead is dissolved in the cytoplasm of the cell. Number 9 says you discover another protein that has an unusual structure. All the hydrophobic amino acids are on the outside of the protein, and all the hydrophilic ones are on the inside. Where in the cell might you expect to find such a protein, and why? Well, this is similar to a problem that I talked about in one of the other problem sets. And now that you've seen how the hydrophobic nature of a membrane can affect the structure of a protein, I think you can get a pretty good idea about where this problem is headed. If we look at a protein that's found in a membrane, for example, we could imagine it might have a structure that looks something like this. So here's our membrane. And I'm just going to draw a small portion of this protein. Let's imagine this protein is this little tube that I've just shown right here. This protein would be existing mostly in a hydrophobic environment because it's touching these long nonpolar tails. We would expect that that interaction would be very good and very easy to form right there because there's virtually no water here, and the hydrophobic portions of the protein would be on the outside. Well, the curious part of this protein is why there are hydrophilics on the inside. And the hydrophilics on the inside tell us that there must be something having to do with an aqueous environment because that's what hydrophilic amino acids like. Well, the hydrophilics on the inside, of course, are a place where water can interact. And I, in another problem set, talked about the protein porin. Porin has a structure like this. It's found in a lipid bilayer. And water actually flows through it. So if I'm going into a cell from out of a cell, or the other direction, it doesn't really matter. Porins allow the movement of water in both directions. We could see that the porin, having the water associating with the inside of the membrane, would be interacting with amino acids that have folded on the inside. And the outer part of the porin would be associating with the long tails of the lipid bilayer. So this porin is really an inside-out molecule because partly of its function, carrying water, and partly because of the environment in which it's found in the nonpolar part of the lipid bilayer. Okay. Number 10 says you discover a new protein that can only be denatured when a chelating agent, such as EDTA, is present, what would you conclude about its secondary structure? Well, that, first of all, is wrong. That should say tertiary. And that was a typo that didn't get corrected in the first place, and it'll be corrected in the final one. What would you conclude about its tertiary structure? Well, what you would conclude about the tertiary structure of a protein whose tertiary structure was destabilized by a chelating agent would be that, in fact, the material that's being chelated is essential for the tertiary structure of the protein. A chelating agent is going to take away or hold on to a metal. So, for example, EDTA is a chelating agent that is able to bind to magnesium very readily. So in this case here, we might say that, well, one of the things that we might expect would be that the protein that had this property, in this case, might have had magnesium or some other metal ion that could be chelated or bound by EDTA. RNase, it says, can't be easily denatured unless a reducing agent, such as mercaptoethanol, is present. Interestingly, it can be renatured, but only to a limited extent. The percentage of renaturation can be increased by addition of a tiny amount of mercaptoethanol. Explain 2 reasons why this might be the case. Okay? So this is an unusual thing, a very unusual observation. In class, in the lecture, I talked about the fact that RNase is a protein whose tertiary structure is very stable. It's very difficult to denature and inactivate RNase by heating, or simply only by heating. And this is because RNase has a lot of things that help it to stay stable even at very high temperatures. RNase, for example, has disulfide bonds that help hold the protein together, and that the boiling and other treatments to the RNase won't denature it. Now, many proteins have disulfide bonds that help hold them together, but RNase appears to have specific disulfide bonds that are critical to the overall structure. In other proteins, they may not hold enough of the protein together to allow it to be stable at a high temperature. Most proteins are not stable at a high temperature. If we want to denature RNase, the way to do it is to treat RNase with a chemical that destroys the disulfide bonds, and then boil it or add urea or some other agent that causes the hydrogen bonds to be broken. Well, one such agent is mercaptoethanol, and when we add mercaptoethanol and then we either add urea or we boil the RNase, we discover that we can completely denature it, and if we measure the activity at the high temperature, we see that, in fact, RNase is not active. If we allow RNase to come back to its regular temperature, or if we've used urea, if we get rid of the urea, what we discover is that a small percentage of the activity that RNase had will be restored. Now, this is very critical because it tells us that the sequence of the RNase protein is sufficient to help the protein to cause it to refold properly and restore its activity. Now, it tells us that we don't get all of the activity back, so that only some of the activity is regained. The problem says that if I add a tiny amount of mercaptoethanol to the solution as it's renaturing, that I can actually get more renatured. I can get more activity than if I don't add the mercaptoethanol. Well, that seems a little contrary because you remember that mercaptoethanol was helping to break those disulfide bonds. So the problem says give at least 2 reasons why this might be the case. Well, the answer lies in the fact that there are a lot of incorrect disulfide bonds that RNase can make. Normally, as RNase is being synthesized, the disulfide bonds occur during synthesis. So that, for example, 2 sulfurs that are being synthesized might form a disulfide bond very readily, and other sulfurs that are very far away on a cysteine wouldn't appear because they hadn't been synthesized yet. However, if I take a full RNase and I take it apart and I allow it to come back together, now every possible sulfhydryl on a cysteine could, in theory, come back and interact and cause disulfide bonds to form that would not be stable or not give a stable final structure. So if we add a small amount of mercaptoethanol, it can help to keep double bonds that, for example, are incorrectly being made to come apart and allow the protein to properly fold so that the correct disulfide bonds can be made. So the small amount of mercaptoethanol actually facilitates the proper folding and the proper alignment of the sulfhydryls to form disulfide bonds and make a functional RNase. The second answer to that question is that if we think about an RNase chain as being one of the chains right here, and we think of a separate RNase chain being over here, each of these have sulfhydryls sticking off of them. And since these are floating around and bouncing in solution, this one can bounce into this one and hit, and disulfide bonds can occur between the individual chains. When this occurs, of course, the proper folding cannot occur at all. And as a consequence, RNase would be non-functional. Again, a small amount of mercaptoethanol will help them to come apart